And just when you thought the Calvin cycle was going to be easy, here it comes. There are kind of different versions of the Calvin cycle. And this is video 9.5 about the C3, C4, and CAM versions of the Calvin cycle. This video will cover these unit goals. Press pause if you want to check them out. To appreciate why we need these modifications for the Calvin cycle, it's important to consider the struggle that land plants have with keeping enough water to stay alive. Remember, water is a reactant for photosynthesis, so without water, a plant can't do photosynthesis, and without photosynthesis, a plant can't live. For land plants, when the next water will come can be unpredictable. When's it going to rain again? So plants have evolved some special adaptations to minimize water loss if they live on land. One of these adaptations is a waxy cuticle. The waxy cuticle is on the top surface of the leaf, superficial to or above the upper epidermis. The cuticle is a hydrophobic material, which essentially creates a seal to keep water from evaporating out of the top of the leaf. If water tries to evaporate out of the top, the water's going to run into this hydrophobic waxy cuticle, and the buck stops there. The stomata are the holes in the bottom of the leaf that allow the carbon dioxide to come in and the oxygen to go out. Well, water vapor can sneak out of those stomata too, so the guard cells that flank the stomata have evolved to be able to close the stomata when water levels are too low. And even the very fact that stomata are concentrated on the bottom of the leaves of land plants is an adaptation against water loss as well. If stomata were on the top, so much more water could evaporate. So, plants can close their stomata when water levels are low. This is a good thing to conserve water, but closing stomata has disadvantages as well. First, and most obvious, when the stomata are closed, CO2 can't diffuse in. And on top of that, the CO2 is being used up in the Calvin cycle, so the levels of carbon dioxide end up going down. Moreover, the waste product of oxygen can't escape, and so oxygen levels build up. This doesn't seem like a big deal, but a flaw in Rubisco makes this a big deal. Remember, Rubisco stands for ribulose, bisphosphate, carboxylase, oxygenase. The carboxylase activity of Rubisco is the carbon fixation step in the Calvin cycle. It's the oxygenase activity of Rubisco that's a problem. You see, Rubisco can attach either carbon dioxide or oxygen to RUBP, and that's a big problem. When carbon dioxide and RUBP are the substrates, it's business as usual. The plant is happy and efficient when carbon dioxide acts as a carboxylase and fixes carbon in the Calvin cycle to make G3P. But when oxygen levels get too high, Rubisco can grab oxygen instead of carbon dioxide and fix oxygen to RUBP. When this happens, the 5-carbon oxygenated RUBP will split into a 2-carbon and a 3-carbon piece. The 2-carbon piece ends up splitting into 2 single-carbon carbon dioxides. But then the carbon dioxides just end up going back into the Calvin cycle to make RUBP. So when Rubisco acts as an oxygenase, the process uses NADH and ATP, but doesn't make G3P. Basically, the plant is just wasting ATP and NADPH, making photosynthesis a lot less efficient. The process of fixing oxygen to RUBP has a special name: photorespiration. And photorespiration makes photosynthesis less efficient, and that's a bad thing for plants. Here's a graphic to show you how Rubisco is a carboxylase and oxygenase. The left side of the diagram shows Rubisco carboxylating RUBP. RUBP plus carbon dioxide. And this is carbon fixation, phase 1 of the Calvin cycle, that results in the generation of G3P, which is just shown here as a generic carbohydrate. The right side shows Rubisco oxygenating RUBP, RUBP plus oxygen. The result is a 3-carbon piece and a 2-carbon piece. The 2-carbon piece gets broken up into carbon dioxide, and the 3-carbon molecule just gets recycled back to RUBP. The oxygenation gives products that use NADPH and ATP, but don't give any useful products. Rubisco, as an oxygenase, is a problem. But how did this problem arise in the first place? Well, it's thought that when Rubisco first arose, the conditions of Earth at the time didn't really create any type of aggressive selection pressure against the oxygenase activity of Rubisco. Now, however, plants that live in hot or dry conditions have had the need to evolve alternate routes to fix carbon. The alternate pathways use workarounds to concentrate carbon, shielding Rubisco from high oxygen exposure. The adaptations to reduce photorespiration are called C4 photosynthesis and CAM photosynthesis. C4 photosynthesis relies on separating carbon fixation in the Calvin cycle into different cells. CAM photosynthesis, on the other hand, relies on separating carbon fixation in the Calvin cycle into different times of the day. Let's go into more detail about plants that use the C4 strategies for photosynthesis. Some examples of C4 plants are some types of grasses and corn and sugarcane. The main things that are different about C4 plants are that C4 plants have different leaf anatomy, and C4 plants first fix carbon with a different carbon fixing enzyme called PEP carboxylase. Let's see how C4 plants fix carbon. What we've considered normal leaf anatomy so far is actually what we would call a C3 plant. As we've already learned, bundle sheath cells surround the vascular bundle consisting of xylem and phloem. In a C3 plant, or normal plant, bundle sheath cells don't have chloroplasts. Also, mesophyll cells in a C3 plant are more specialized, where palisade mesophyll cells are along the top of the leaf where the sun shines, and spongy mesophills are along the bottom. In C4 plants, there are a couple of tweaks to the system. First, notice the difference in bundle sheath cells. In C4 plants, bundle sheath cells do have chloroplasts. Next, check out the mesophyll cells between the two. In C4 plants, rather than being specialized, C4 mesophyll cells are more uniform and look really similar to one another. But the mesophyll cells do pack really tightly against the bundle sheath. The secret is that the bundle sheath is where most of the Calvin cycle will happen. So the bundle sheath is where most of the Rubisco is. So then making a tight seal around the bundle sheath keeps the bundle sheath shielded from the areas of high oxygen, which would be these air pockets out here in the leaf, thus reducing the chance of Rubisco doing oxygenation reactions. Let's check out the carbon fixation workaround. In a conventional C3 plant, light-dependent reactions take place in the mesophyll cells on the thylakoid membrane, and carbon fixation happens in the stroma by Rubisco as phase 1 of the Calvin cycle. What's different about C4 plants is that C4 plants do light-dependent reactions in mesophyll cells and bundle sheath cells as well. Carbon fixation, however, happens in a different place. Mesophyll cells are still in charge of carbon fixation, but mesophyll cells fix carbon in their cytoplasm rather than their stroma. Carbon fixation doesn't happen with Rubisco, but it happens with the enzyme called PEP carboxylase. Once carbon is fixed in C4 mesophyll cells, the carbon is passed from the mesophyll cells to the bundle sheath cells, from here to here. Once carbon is passed to the bundle sheath cells, the carbon dioxide can now enter the Calvin cycle when Rubisco takes that carbon dioxide and attaches the carbon dioxide to RuBP. Now, if you're not comfortable yet, don't worry, we'll examine these reactions in more detail. Well, let's take a look at this PEP carboxylase enzyme. We know that PEP carboxylase fixes carbon in C4 plants, but this enzyme also fixes carbon in CAM plants too, so pay close attention. Like the name suggests, PEP carboxylates PEP. PEP is a 3-carbon molecule, and PEP carboxylase adds the 1-carbon carbon dioxide to PEP. And that results in the generation of a 4-carbon piece. This 4-carbon piece right here is how C4 plants get their name. This 4-carbon piece is why we call C4 plants. Remember, PEP carboxylase is found in mesophyll cells of C4 plants and not in C3 plants. So in C4 plants, PEP carboxylase only works to fix carbon in the cytoplasm. Let's fold all of these pieces together and tell this C4 story in its entirety. What we're looking at here is a leaf in a C4 plant. You can see those bundle sheath cells with chloroplasts, and you can see these mesophyll cells tightly wrapped around the bundle sheath cells. Over here, we're zooming in on one mesophyll cell snuggled really close to a bundle sheath cell. So the mesophyll cell takes up carbon dioxide from a nearby airspace. That carbon dioxide is attached to PEP by PEP carboxylase to create a 4-carbon molecule. That 4-carbon molecule is then passed from the mesophyll cell into the bundle sheath cell. Once in the bundle sheath cell, the 4-carbon molecule drops off 1 carbon as carbon dioxide. That carbon dioxide is able to enter the Calvin cycle when Rubisco adds the carbon dioxide to RuBP. The 3-carbon piece just heads back over to the mesophyll cell to pick up another carbon dioxide later. I know this seems a bit weird, but having this set up isolates Rubisco from the areas with high oxygen. Rubisco is at its highest levels in these bundle sheath cells during the Calvin cycle, and oxygen is separated over here at its highest levels in these air spaces. The mesophyll cells being in between keeps these two areas separated and reduces the chance for photorespiration. So plants with the C4 adaptations have a selective advantage. Separating Rubisco and high oxygen levels reduces wasteful photorespiration, but there's also a cost. Sending the carbon from the mesophyll cells to the bundle sheath cells and back costs some ATP. But the ATP from moving these carbons back and forth costs less than the ATP and NADPH cost of doing photorespiration. Plants called CAM plants have found a different workaround to Rubisco's oxygenase activity. CAM stands for crassulacean acid metabolism. That's why we call them CAM plants instead. And CAM plants have adaptations to survive their hot, dry climates. What CAM plants do that's different is open their stomata at night to fix carbon using PEP carboxylase. CAM plants build up carbon molecules in their vacuoles at night. Then, during the hot daytime, CAM plants close their stomata and gradually release the carbon that they've stored. Some examples of CAM plants include succulents, cacti, pineapple plants, and orchids. This diagram really lays out the CAM strategy nicely. Let's start with the nighttime half. At night, CAM plants open their stomata. Since it's not hot, the CAM plants won't lose as much water as if they had their stomata open during the day. When carbon dioxide comes in those open stomata, the mesophyll cells fix carbon dioxide using PEP carboxylase. PEP plus carbon dioxide gives a 4-carbon molecule. The 4 carbon pieces are moved into and stored in the vacuole of the plants. 4 carbon pieces are made all night long and build up in the vacuole at night. Once the sun rises, the stomata are closed to minimize water loss. But now carbon dioxide can't come in. But it's not that big of a deal because of what happened at night. What CAM plants do during the day is transfer these 4 carbon pieces that were built and stored at night back to the chloroplasts from the vacuoles. Then, these 4 carbon pieces release a carbon dioxide molecule, and that carbon dioxide molecule is taken by rubisco, added to RUBP, and the Calvin cycle can happen. This 3 carbon piece will just be turned back into a 4 carbon piece at night. So this is the workaround for CAM plants. So let's recap. Because water availability can be an issue for land plants have special adaptations to deal with potential water shortages. Land plants have waxy cuticles on the top of the leaves and closable stomata concentrated on the bottom of the leaves to reduce water loss. When leaves close their stomata, this can reduce water loss but cause oxygen to build up in the leaves. This is a problem because rubisco can wastefully attach oxygen to RUBP instead of carbon dioxide and reduce photosynthesis efficiency. When rubisco attaches oxygen to RUBP, that process is called photorespiration. Photorespiration is a bad thing. Some plants, called C4 plants, have evolved strategies to overcome photorespiration by fixing carbon in mesophyll cells, passing that carbon to rubisco that's isolated in bundle sheath cells. Rubisco in high oxygen areas are isolated by different anatomy. CAM plants are different types of plants that use a different strategy to overcome photorespiration. CAM plants overcome photorespiration by fixing carbon at night and using up that carbon during the day. In other words, rubisco is separated from high oxygen conditions by using a different time of day strategy. Be sure to ask questions in class if any of this was confusing for you.